Hi there, my name is Justin Day. Today I'll be discussing the results of my research project titled Arbuscular Mycorrhizal Fungi Colonization Decreases Under High Precipitation and Compost Treatment in Semi-Arid Rangelands. Arbuscular mycorrhizal fungi, which I'll abbreviate as AMF, are a form of symbiotic fungi that colonize the roots of 80% of vascular plants. And in this relationship, they increase nutrient acquisition from the soil to the plant, while simultaneously receiving carbon from the plant. Because of this relationship, they're proposed to be important for management in agricultural systems. And one agricultural system of interest are Californian semi-arid rangelands. Here, compost application has been proposed as a practice to increase soil nutrients to the plant. Simultaneously, semi-arid rangelands are characterized by high rainfall variability and frequent drought. However, the compounded effects of compost and high rainfall variability on the AMF-plant relationship are largely unknown. So here, we will measure the AMF-plant relationship under fertilizer and compost amendments across a precipitation gradient in semi-arid rangelands. We expect that as increased nutrients and precipitation supplement the benefits of AMF colonization, plant hosts will reduce carbon delivery to AMF. Specifically, the first hypothesis is that AMF colonization rates will be highest under no amendment soils, they will decrease under inorganic fertilizers, and be lowest under composted conditions, and these effects will be exaggerated by increased precipitation. Second, plant root biomass will negatively correlate with AMF colonization under soil amendments. Moving on to our methods, we used a full factorial design consisting of 3 nutrient treatments: compost, fertilizer, and no amendment, and within each of those we had 3 precipitation treatments: drought, ambient, and high precipitation. From each plot, we collected soil samples, and from each soil sample, we collected root samples, which were then cleared using potassium hydroxide and stained using an ink vinegar solution to visualize the AMF structures, which can be seen in the figures on the left. Using these samples, we were able to quantify AMF colonization as a percentage of root length. If we move on to our results, this first figure shows us AMF colonization across the different treatments, specifically compost, fertilizer, and no amendment treatments from left to right, and within each of those facets we have drought in red, ambient in yellow, and high precipitation in blue. We can see from the far left blue column that high precipitation and compost treatments yielded the lowest AMF colonization. Conversely, the next figure is set up in the same way, but shows us plant root biomass. Here we find that plant root biomass was highest under the exact same conditions that colonization was the lowest, with compost and high precipitation. The third figure shows the relationship between AMF colonization and plant root biomass across the 3 different nutrient treatments. Here, we found a significant negative relationship between AMF and plant root biomass under compost treatments. With relation to our hypotheses, our first hypothesis was not fully supported by the AMF colonization data, as there was no significant relationship between nutrient treatment alone and AMF. However, the combined effects of nutrient treatment and high precipitation do partially support this hypothesis. Second, our second hypothesis was partially supported by the negative relationship under compost conditions, but was not by the lack of relationship under fertilizer conditions. Taken together, we can consider these results in a cost-benefit analysis from the perspective of the plant's investment into AMF, where the cost is carbon delivery to the AMF, and the benefits, which are not all explained in this paper, include nutrient acquisition and drought resistance, just to name a few. In low nutrient availability conditions, we have a high relative benefit of investing to AMF. However, if we supplement this low nutrient availability with compost or fertilizer, we decrease the benefit of AMF and increase the relative cost, which could explain the divestment of carbon to AMF structures and instead into plant root structures, as seen in the third figure. Taken together, these results provide a basis for understanding the AMF-plant relationship and how dynamic it is under compost treatments and high variability rainfall in the specific context of agricultural systems.